Good afternoon, both Dr. Bala and Dr. Chakravorty, the chairperson of this session. Also, thanks to the today the inviting me for this occasion of the organizing committee. And thanks to all audience, virtual audience, those are here in the connections of virtual meet. I am going to present my screen share to start my lectures. The topic which today I am delivered here, the tissue culture of medicinal plants for production of bioactive compounds. This is my topic. Before going the detail of the tissue culture, I just mentioned some known facts of the medicinal plants. Medicinal plants is a power of ancient times, all of we know. And it's a huge pharmacological properties due to its secondary metabolites, which act as an added value in the bioactive compounds. And all of we know the secondary metabolites, the comparatively complex structures, and produce a response to various forms of stress, for both the abiotic and abiotic stress. For their defense mechanism, they reduce the secondary metabolites. Thus, secondary metabolites or bioactive compounds is widely used in the pharmaceutical industries, cosmetics, dietary, flavored and dyes, etc. In different, all of we know the facts. But I just mentioned here, before going to cultivation or business or whatever may be, the quality of plant material is very important for medicinal plants. So quality assurance all growers should take attention in the quality assurance. The initially first two part is very important, like botanical assurance and as well as chemical characterization of the medicinal plants. Proper identification of medicinal plants is very important. Otherwise, within the same genus, lots of species are there, and there is the difference is very little bits. So experts can identify, particularly those authority of the Botanical Survey of India or other herbariums can identify the subspecies or variety, whatever may be, that should be a very important for before going to the cultivation. And another important is the right clone or right chemotype. That's called, we tagged as the elite genotypes or elite chemotype. The highest percentage of active compounds on secondary metabolites is from an individual's genotypes is important. That should be characterized from a recognized laboratory which clone is better to cultivate as a starting material. Without these two, I think no one should start the cultivation of the medicinal plants. Look, one example I just cite here, category 1 medicinal plants produce raw material 100 gram of tissue. If there is a 1 gram per 100 gram, the metabolite contained, and selling price of the 100 rupees. Whereas in the biomass, category 2 is a biomass is used nearly five times more, but the content is low. That is the 0.1 gram per 100 gram, and they will earn only 50 rupees instead of 100 gram, although the biomass is less. So now I'm here I am showing the plant materials. The less biomass also an important if their content is high. So before going to start the material cultivation of the particular cultivars, you should ensure the both things as a correct identification as well as the content of the bioactive or secondary metabolites. There is some factors. I think there is some factors. One is the intrinsic factors, that is the genetic factors, genotypes, or chemical factors, that is chemotype is also an important aspect. And another is the extrinsic factors, that is the environment, ecological connection, cultivation, harvest, post-harvest processing, storage practice, all are extrinsic factors. So these are very important. We are I gave you one example in the Calmec or Androgophis paniculata. We are screening the elite plant material within the West Bengal. We are harvested from nearly 70 localities of different districts of West Bengal, and andrographite content varied from 0.2% to 5.5% in different localities as well, different times. So before going that, this screening for selections of elite clone is very important. Another important aspects which I mentioned here, that is the particularly those that are particularly the conserve. We should conserve the selected elite genotypes or chemotype for future use, and should identify the best possible environment where plant can synthesize more plants material, both in quality and quantity. So in both are, we are recommended these two important aspects. And now question is, why seed-derived plants are not fit for quality assurance? That's very important. Because seedlings plants are not true to type to mother plants. Once we selected the elite chemotype from an individual plants or a group of individual plants, if we germinate it to produce the successive generation, they segregates their trait through the Mendelian genetics pattern, and they lost their stability or same quality assurance with the mother plant. So heterozygosity is the main cause of this seedling plants. And another is the no assurance about the genetic purity of the offspring or seedling. So very difficult to retain the superior quality in the seed-derived plants. So it is an important issue for those farmers are used for the seeds for a mass propagations of plants. So there is some source of genetic natural variation. One of the important aspects is the crossing over or genetic recombination during the meiotic process, where the random reshuffling of the segments of the genes, and another is the variation is contributed to the random fertilization. So the seeds are failed to produce the same quality of plants in their successive generation. And here you see the crossing over can change the genetic alignment. From the old time, I think from the ancient time, without the knowledge of genetics, the peoples are now know how they cultivate the mango plants. Mango plants never see the propagate through the seedling. They choose the grafting or cuttings or whatever may be for their purposes. So this is the only cause. The vegetative propagation can solve the quality assurance problem due to the effort of genetic recombination. So it is an important issue of today that the seedlings have lots of problem. So in nature, in common practice in our society, the cultivars, they use the stem cutting, grafting, root cutting, and so on for the vegetative mode of propagation. Even some bulbaceous plants, sucker, rhizome also used for propagation to avoid the genetic recombinations or variation problem. But this genetic variation is surely 100% checked through the plant tissue culture system. And plant tissue culture is nothing but a simple vegetative mode of propagation. So what is the problem? Some people can raise the question, why we go to tissue culture when the sucker, rhizomes, cuttings, graftings are there? Then why we spend more money and more special facility for tissue culture, right? The sucker, cutting, grafting, both are same genetic or basic mechanism is same, but there is a criteria of the number of propagules production. In cuttings, grafting, from one single mother plants can produce 2, 3, 4, or maximum 10 plants. But tissue culture-based plants can produce the huge number of same quality of plants, of clonally propagated plants, more than 8 to 10 million plants per year from just a 10,000 square feet floor area. If you arrange 1,500 floor area and 6 storage building, you can produce at least 10 million plants per year. So huge amount, same quality of plants you can get through the tissue culture procedure. If you produce the viral elimination is a great challenge in the disease-free plant. So it can easily eliminate through the meristem culture, through tissue culture technique. And the phytochemicals of secondary metabolites, you select the particular elite clone from single clone. You can propagate the particular chemotype in the tissue culture methods. Another important thing is the generation of polyploid plants in the in vitro condition, that is tissue culture methods, can be achieved. And the elite genotypes or elite chemotypes can be conserved. They can easily avoid the natural digester through the in vitro test tube plants. So, and there is no seasonal barrier. The medicinal plants can grow throughout the year within an environmental condition. And it is the basic idea of how the plants from to the mixture of secondary metabolites, single target compounds, and this one, the steps of particularly this 5 steps we consider for selection of elite genotype, collection, extraction, quantification, screening of elite chemotypes, and after screening, the propagation of large-scale propagation, mass propagation, clonal propagation in tissue culture. This is the basic patterns of this method. We just show you the steps of extraction and quantification of elite clones. The pharmaceutically active compounds, all of we already have mentioned here, that the secondary metabolites and that this secondary metabolites can synthesize within the glass vessels or culture vessels around the year. Both their quality and quantity both are maintained. Look here, there is the asparagus racemosas. You see within the 400 ml jam bottles has produced more than 250 microshoots from a single vessels, and you cut the individual microshoots to induce a root, and you can get such a type of complete plantlet in the tissue culture system. So from the 250, more than 250 shoots can produce 250 individual plants within a small space. You see here, we also propagate, I just go through the first, the Bacopa, or that is the Mentha piperta. You see, we standardize the aloe vera, is huge number of aloes produced in our lab. And whether it is the genetically stable or variant of the somaclonal variation, that we can check through their 3 parameters. Number 1 is the karyotype patterns, both in the somatic chromosome as well as meiotic behavior, followed by our molecular markers of the RAPD, ISSR fingerprints through their genetically stable or their variant, and even the secondary metabolites of aloe. We also propagate the huge amount of artemacin synthesizing plants, that is Artemisia annua. All of you know the only Artemisia annua can synthesize such a type of arteether and sesquiterpene of the artemisinin for the treatment of the malignant malaria of the throughout the world, that can easily propagate in the tissue culture technique. And the tissue culture rest plants are transferred in our polyhouse and shed net house to grow in the potted condition. All are tissue culture rest plants. And you see here, we also cultivate, generate the huge number of ashwagandha or Widania somnifera. After regeneration, we are transferred in the field condition to maintain or check the secondary metabolite contained. And that also we check through the widaferin A and widanolate through our HPLC and other chromatographic methods. We also develop our system in our lab with the Plumbago zeylanica, particularly for plumbagen production, both in the tissue culture system as well as the cell culture technique. And you see here the stevia very nicely propagated, multiplied in the in our lab in the culture I think in the culture vessels with a huge number of multiplication, large scale production of stevia. But there is some drawback in our cases. The good clone are not available in normal courses. So if we receive or collect or manage some good clone from any sources, we can propagate huge number of plants from a small space with the maintaining the quality. We also propagate the different clones of ginger for ginger oil purpose. So after screening, we propagate the elite clone in vitro condition. You see here, for vanillin purpose, vanilla is also propagated in my lab. Centella asiatica, even in the large cardamom, also multiplied in the lab condition in my lab. And another, Campharia galanga, the kangi, is also growing huge amounts. And in the field condition, we transferred huge amounts in the field condition. And it is another species of Campharia angustifolia. We are propagate from in vitro condition, tissue culture to the field condition, and we check their the essential oils through the GCMS chromatography. Not only the shoot or meristem culture to produce, it is the Indian sequel. We use a small part of the leaf. You just see the cut and produce a huge amount of somatic embryo. And individual embryo can regenerate a complete plant. You see is a lamina, leaf lamina. We prick some area of the lamina, and a huge number of individual somatic embryo is induced. And individual somatic embryo, you see here the initiation of shoot birds are started from these places, induced from these places. So from a single leaf, you can get 5,000 to 6,000 plants in a 3 months of the Argenia, that is Indian sequel plants in our laboratory. You see, even we can use some prick or wound response in the lamina, and within the 15 to 21 days, 3 weeks, every prick induced individual somatic embryo, and individual somatic embryo ultimate regenerate complete plants like this. You see here the complete plants with the bulb, with the root system and leaf system. Huge number of plants are growing in the in the liquid flask culture media. Another thing is another aspects of the synthesis or induction or enhancing the synthesis by elicitation of the elicitor. We treat the elicitors of both in the biotic elicitor and abiotic elicitors, which can induce through their signal, use as a signal molecules through their signal transduction pathway. They are used as a defense because the plants start their defense mechanism for in more biosynthesis of more active compounds. You see, it is the basic patterns of the how they particularly compounds elicitor and act of the there is a elicitor says act as a ligand, and there is the membrane is the receptor proteins. They go to signals in the transcription factors. They change some mRNA expression levels, which can synthesize more enzymes for secondary metabolite synthesis. Cell culture, instead of complete plants, only cells can synthesize the secondary metabolites, and this can grow in without that depend on any environment. That geographical and seasonal variation, they are independent. So cell culture is another aspect of secondary metabolite, which we are commonly practiced in our lab. You see here is that a segment of the leaf. From the segment of the leaf, the mature leaf, I think there is a highly differentiated structure is converted into undifferentiated mass of cell that is called the non-morphogenic, that is called the callus, undifferentiated mass of cell. Here is a see after subculture, successive subculture, this such type of callus that grow in a culture flask for conical flask. And this every tissue or cells can synthesize the secondary metabolites either in normal condition in the normal culture media, which is standardized, or we can induce more synthesis by different types of biotic and abiotic elicitor to trigger their defense mechanism. And after that, this callus is transferred within the liquid media. It is a suspension culture of liquid suspension culture of a particular species, plant species. And you see the single cells is here and divide within the suspension system. So cell culture is also an important to synthesize the secondary metabolites. So there is no need of plants. There is no need of field. They are only synthesize the secondary metabolites in the flash condition or in a large scale of the bioreactors or fermented. They can synthesize the secondary metabolites from the individual cells. So it is a less costing. I think there is a less time is required for the suspension or cell culture technique. The phosphorylin is another diterpene, all of you know, from the Coleus phosphorylin. We developed without the complete plants. We only developed the root culture system. I think all of you know the site of synthesis of the phosphorylin in the root system. So why we should go to complete plant? Better to only root culture can maintain, and roots produce a huge amount of phosphorylin of diterpene instead of leaf and shoot spot. So this is another approach to produce the secondary metabolites or bioactive compounds in the root system, and root is the nice method system to synthesize the particular compounds. So you see here, this is the root culture system, and if we dry the root is like that, huge amounts of roots come out from the flask. And also the Coleus phosphorylin, we select the particular cell lines for a selection of elite cell line for the synthesis of phosphorylin purpose. And another is we also developed the crown gall system through the infections of the agrobacterium mediated transformation for synthesis of crown gall for synthesis of secondary metabolites. And the last part which I mentioned here is the in vitro conservation. I think there is another challenge. I think today a large number of species are verged to extinct or going to extinct due to overexploitation or destructive harvesting or maybe poor seed setting. So the germplasm conservation in vitro condition in tissue culture system is more efficient and more important. They avoid the natural disasters. They can easily maintain for 10 to 20 years in a lab condition. In vitro conservation have some advantage: sustainable utilization of genetic diversity. Different elite clone can germ preserved in the laboratory condition, and this system is considered as an ex situ conservation of plant germplasm conservation. Is here. This is the Glorious superba synthesized highly costly. I think costly secondary metabolites alkaloid is the colchicine. All of you know the different types of products are now available in colchicine. Initially, it's considered as the anti-gout drug, but last 2 or 3 years, there is several reports in the cardiology journals that there is a minor amounts of colchicine also act to avert the cardiac arrest. So minimum 0.1 mg probably, I think, I just remember the amount of injections can help. Our tablet is now also available in India, the colchicine containing, and it is synthesized. Initially, we think we know that the colchicum maternal is the sole source of colchicine, but now it is now established that the Glorious superba, basically flame lily, is known to as ornamental plants, but they also synthesize the more amounts of colchicine than colchicum autumnal. And this plant is now red listed by IUCN as a threatened plants of these plants. And you see here the list of threatened species, and there is a number of IUCN number is here. And we can establish the in vitro system or tissue culture system to propagate huge number of glorious superba in the culture system. And these plants also synthesize the colchicine in their rhizomatous part. You see the in vitro condition, rhizomes are developed within the in vitro condition. You see here the root system also develop and we transfer to the field condition. So this is another way by which we can conserve the elite genotypes or threatened plants in vitro condition, which is not possible or propagate properly in the field condition. Sorry. That is another method by which we can conserve our elite crop. That is the artificial seed. Initially, I start my lectures with the botanical seeds which are developed after fusions of the two gametes after sexual reproduction, but it is the artificial seed or synthetic seeds which avoid both the fusions or fertilization process in the plants. It is totally synthesized this seed in the in vitro or laboratory condition. It is developed after the encapsulations of by alginate bits of the in vitro tissue culture derived somatic embryo are encapsulated within the alginate bits with the endosperm of the media is used for the nutrient purpose. So this is the method. It is a method of the synthetic seed. We can encapsulate it or envelop the somatic embryo within the bit of encapsulated bit, and within the bits there is a nutrient. There is a synthetic seed coat, synthetic embryo, that is somatic embryo, and which are endosperm, which provider of the nutrient also. We serve as a nutrient, as an artificial nutrient media. So this concept also help, these types of culture can help to conserve or propagate in both purposes, but through the vegetative mode of propagation. This artificial seeds are true to type of the source plant. There is no variation, and those are not. There is some advantage of the synthetic seeds. Also, that is the basic concept. It is the synthetic seeds germinate in the field condition or in the laboratory condition, or just there is another approach for the synthesis of secondary metabolites is through the plasmid and through the genetic transformation. You see here this within it is within a somnifera. Roots are developed within the lamina of the leaf by the insertion of a particular gene. You see here that from the leaf, huge amounts of hairy roots are developed within the leaf, and this root can be cultured without the growth regulators. And we have confirmed the foreign gene is present or absent through the southern blotting, by southern hybridization, as well as PCR through the particular primers. And this is the content we also measured, and we are finally the maximum plants are transferred in the initial in the polyhouse and subsequently in the greenhouse. And thank you, sir.